For this research project, we decided to study the effects of water quality on the biodiversity of algae. Based on previous research, we hypothesized that there would be a positive correlation between algae biodiversity and water quality characteristics. In order to do this, we looked at 3 rivers, Grand River, Welland River, and Looking Glass River. Within these rivers, we collected data for water temperature, river speed, pH levels, phosphate levels, and algae biodiversity over a period of 3 weeks. A correlation test was conducted between algae biodiversity and the 4 variables separately for each river. The results from these tests suggest that there was not enough significant data to show consistent correlations between water quality characteristics, phosphate and pH levels, and algae biodiversity. The lack of correlated data in this study is likely due to the fact that it was completed during a low algal season, thus providing us with limited access to the growth and biodiversity of algae. Along with this, the limited timeline only provided a small understanding of the water quality trends for the rivers as well. In the future, this experiment may be repeated in an alga abundance season with more variables, higher quality resources, and a more spread out timeline. We decided to further research on if algae growth and biodiversity is impacted by the decreasing water quality observed between urban and rural rivers. It is common knowledge that the human population has displayed negative effects on algae populations within rivers. Aquatic health can be influenced by fertilizers, industrial pollution, animal waste, and sewage. Rivers in urban areas are also known to have higher levels of phosphate and nitrogen, which can eventually lead to degeneration of ecosystems and a general decline in the ecological status of the fish population. This research is very important to our society because it can further our knowledge on the effects of algae on ecosystems. This research can also allow us to develop better river management plans in order to accommodate for the effects of algae growth on certain rivers. Our null hypothesis for our research question was that there would be no correlation between water quality and biodiversity of algae, and our alternative hypothesis was that there would be a correlation between the water quality and the biodiversity of the algae.
3 rivers were sampled to gather this data. These 3 rivers were the Welland River in Pelham, Ontario, the Grand River in Lansing, and the Looking Glass River in DeWitt. At each river, 3 locations were established with one center location, 1 kilometer upstream from the center, and one location 1 kilometer downstream from the center. At each location, 3 samples were taken. This was done 4 times for each river, totaling 36 samples at each river and 108 samples overall. Water temperature and the surface speed of the river were measured at each location on site using a thermometer and a combination of a ping pong ball measuring tape and a stopwatch. Each sample was also tested for pH, phosphate levels, and algal diversity after sampling using pH strips, a phosphate test kit, and an electronic plug-in microscope. In order to figure out what species of algae are being observed, an online database for algal identification from the Canadian Agri-Environment Services Branch was used to progressively find the species being observed and counted, such as Crucoccus and Microcystis. To find biodiversity from algal diversity, a Simpson's diversity index was used. Once all of the data was collected onto a spreadsheet, a correlation test was performed between the biodiversity and the other 4 parameters. These correlation tests also gave the necessary p-values for the data sets.
In order to best display our results, we decided to make 4 graphs. The first comparing algae diversity to river speed, the second comparing algae to biodiversity to water temperature, the third comparing algae biodiversity to pH levels, and the fourth comparing algae biodiversity to phosphate levels. The 3 rivers were plotted on each graph so we could easily compare results between them. Out of the 12 correlation coefficients we calculated, none of them provided strong enough evidence to support a correlation between algae biodiversity and any of the 4 variables for any of the rivers. Out of the 12 p-values we calculated, only 1 of them provided data significant enough to support a correlation. This 1 significant p-value was for Welland River's relationship between algae biodiversity and phosphate levels.
For all variables in rivers, none of the R coefficients suggest significant correlations. This is what we hope to see for water temperature and river speed, because if they showed a strong correlation, that would mean that the impact of algal growth outside of water quality parameters we were interested in. For pH, we expected to see a positive correlation, and for phosphate levels, we expected to see a negative correlation with algal biodiversity. The only significant correlation was found in the Welland River in regards to phosphate levels, likely due to eutrophication. Grand River showed opposite trends in comparison to the other two rivers, likely due to its urban setting, as seen in figures 3, 4, and 5. We failed to reject the null hypothesis that there is no correlation between water quality and algae biodiversity. Problems that arose during our study include having limited access to supplies and a limited timeline. The timeline for this study only allowed us to collect data on 4 separate dates from each river. Although those dates we collected 9 water samples from each river, 4 dates really only provided us with a limited understanding of the water quality at each location. With more resources, we would have been able to look at other water quality factors beyond pH and phosphate levels. And finally, we restricted water access, which made it hard to access algae in the water, and some of our samples contained no algae at all. In the future, this experiment may be repeated in an algal abundant season with more variables, higher quality resources, and a more spread out timeline. Summer is the most algal abundant season, thus completing the study then might allow us more access to an understanding of algae biodiversity. Better yet, completing this study over the course of a year would be better show the impacts of algal blooms and water temperature. Long-term water quality trends should be studied as they would be more likely to have a stronger impact on algal growth. We were measuring momentary water trends, which may not be likely to have a great impact on the growth of the algae in the same sample because the growth is not instantaneous. Thus, utilizing water quality database that even encompasses more parameters or with access to enough resources to compile one might yield a better study. And finally, our take-home message is that there is not enough significant data to show consistent correlation between water quality characteristics such as phosphate and pH levels and the algal biodiversity.